to today's lecture. So today's lecture is on cytokines. I'm sure we've all heard about cytokines and the cytokine storm. And those of you who are interested to know more have already read up on the topic, just because of the severe disease that cytokines cause in SARS-CoV-2 infection. So these are the learning objectives. By the end of this lecture, you should be able to describe the general properties of cytokines, describe the role of cytokines in the immune system, basically in the immune response. And just to summarize what cytokines do, they play a role in development of immune cells. I'm sure you've already immune cell development through the process of hematopoiesis. And you talked about the different growth factors and cytokines that are important in proliferation and differentiation of these cells to effector cells. We'll talk about what cytokines do in the innate immune responses and also in the adaptive immune responses. We'll also outline some cytokine-related disease processes and then outline the clinical utility of cytokines. Those ones we'll just mention briefly. We'll probably talk more about this when you're in year 4. So cytokines are small protein molecules which are used by immune cells to communicate with each other. So they're basically used for communication from cell to cell, or to activate cells and basically cause cells to differentiate into their effector cells so that they can actually do what they're supposed to do. So they are messenger molecules, are soluble messenger molecules. Most of them are secreted by cells of the immune system, but there are also some non-immune cells that can secrete cytokines. For example, endothelial cells. Some are, so we said that soluble, meaning they'll be found in solution, but some are actually found bound to the cell surface. So there are cell surface membrane molecules or, which would act on receptors that are found on other cell surfaces. For example, the CD40 ligand is a cell surface bound cytokine, as well as the Fas ligand. So for those, usually for CD40 ligands, they would bind to a CD40 receptor on the respective cell, and the Fas ligand would bind to a Fas receptor. Those are usually found on T cells, the ligands. So as I've mentioned, cytokines are involved in development and regulation of the immune system. And as I've mentioned, they're also involved in basically the activation and effector function of innate and adaptive immune responses. So we'll describe some of the properties of cytokines. They have some really distinct properties. One is that they are small molecules. They have a low molecular weight rather. They have short half-lives. So once they're secreted, they're not going to be found in these biological fluids for a very long time. So they degrade very quickly. But despite that, despite the small size and their short half-lives, they're very potent, okay? And case in point, the cytokine storm, just a small amount of these molecules causes very potent effects. And because of that, the expression is tightly regulated, okay? So that is both the secretion and also the different receptors that they act on are very tightly regulated so that we don't get very severe effects of these cytokines. So how do they work? They function by binding to the specific target receptors, which would be found on the surfaces of other cells. And then once they bind to the receptor, They cause activate intracellular signaling molecules, for example, the JAK-STAT pathway or other pathways, which would basically send this signal to the nucleus, and then you get alteration of gene expression. So you get certain genes being activated or some being downregulated, and the specific proteins that those genes code for will either be produced or they'll be inhibited. Okay, so that would be the effect of the cytokine. So the outcome of cytokine stimulation depends on the cytokine and on the target cell. So you can either get cell suppression or cell activation. You can get cell proliferation. So you get that cell now producing other daughter cells, or you can get cell differentiation. So one cell now differentiates into another cell with a different function. For example, you can get now a B cell differentiating into a plasma cell, which now produces antibodies, or you can get cell migration. And this is specifically seen with the class of cytokines called chemokines that would now cause some cells to migrate maybe from the bloodstream into the tissues where these chemokines are being produced during the process of inflammation. So against a chemical gradient, and in this case, the gradient is, or the chemical gradient is caused by the cytokine. So those are basically the 5 effects you have of the cytokine: cell suppression or activation, proliferation, differentiation, and cell migration. So this diagram is just showing us how the cytokines work. So you get an inducing stimulus, which activates a cell. Then you get activation of the gene that codes for certain cytokine. You get the cytokine being secreted. It will bind to its cognate receptor on another cell, causes intracellular signal transduction. And then you get another gene or being activated, and the biological effect, which would be one of these 5 that I've mentioned. So cytokines have several modes of actions. They can either act in an endocrine fashion. So this is why you have your cytokine being produced, for example, by your white blood cells, which are obviously in the blood, so secreted in your circulation, and then they go and act on a distant cell, okay? So just the way hormones act, that's why it's called the endocrine action. So it acts at a distant site. A good example of this is when you have, during an infection, and you have a cytokine like tumor necrosis factor being produced by, for example, by your macrophages, and the cytokine is carried to the central nervous system, where it has receptors on cells of the hypothalamus, and what it does is cause an increase in body temperature. And then the patient who has an infection now presents with a fever. So that fever is actually a response of the immune system through the tumor necrosis factor cytokine. And it's supposed to be a protective effect for us. It tells us that there's some inflammatory process going on. But for that patient or that host, it's a protective effect because usually high temperature inhibits the growth of pathogens and also increases the effectiveness of the immune response. They can also act in a paracrine fashion. This is where you get the cytokine being secreted and it will act on nearby cells, okay? So an example of this would be during a type 4 hypersensitivity reaction, when you get your macrophage presenting antigen to your T cells, and then the T cell produces a cytokine called interferon gamma, which now acts on the nearby or neighboring macrophages to activate them so that they're able to kill intracellular organisms. The other mode of action is in an autocrine fashion. Auto means the same cell, so the cell secretes a cytokine and it binds to like a target receptor on the same cell that secreted it. So for example, when you get activation of T cells, usually one of the main things that happens after antigen presentation to T cells, that the T cells start producing interleukin-2, And that interleukin-2 produced by the T cell can act on the same cell to activate it and probably cause it to proliferate. Okay, so you get clonal expansion. So those are the three modes of actions of cytokines. The other properties, remember I mentioned cytokines have specific properties. One is called redundancy, and this is shown on this picture on the right, where you can have different cytokines all causing similar effects. So why we say this is redundant is because maybe if you wanted to block the effect of that cytokine, so for example, here you have an activated T helper cell, which can produce different cytokines like interleukin-2, interleukin-4, interleukin-5. All these cytokines can activate B cells and cause them to proliferate. So if you wanted, maybe it's a clinical scenario where you wanted to block this proliferation of B cells, if you maybe use an antibody that blocks interleukin-2 or its receptor, this B cell can still be activated by interleukin-4 or interleukin-5. So unless you block all of them, blocking just one would basically be redundant. You would still have the same effect caused by another cytokine, okay? The other effect is synergy. Synergy basically means there's an additive effect. So for example, interleukin-4 in addition to interleukin-5 would actually cause the B cell to switch from the default immunoglobulin production. So usually the default immunoglobulin produced by your B cells or your plasma cells is IgM, but interleukin-4 and 5 would switch this to IgE production, okay? So here I'm just giving an example of actually a scenario where you may want to block a certain cytokine. So for example, in rheumatoid arthritis, which is an autoimmune disease, where you get inflammation, usually in the synovial lining of the joints due to an autoimmune response. So the immune system is attacking the host on tissues. Sometimes they use tumor antibodies or monoclonal antibodies against tumor necrosis factor to try and block this effect, but this doesn't cause complete resolution because you still have other pro-inflammatory cytokines like interleukin-1 that are involved in this disease process. So blocking one doesn't mean you stop the disease process completely. The other property of cytokine is pleiotropism. So plio, means many. So, and tropism just means sort of, I don't know how to call it, likelihood or like attraction to certain cell types. So this just means that one cytokine can act on many different cell types. So an example given here is interleukin-4. It can cause activation, proliferation, and differentiation of your B cells. But it can also cause proliferation and differentiation of T cells and also mast cells, okay? So you have one cytokine having effect on more than one cell type. It means all these cell types have a receptor for interleukin-4. So a clinical example of this is a case where you may want to use a cytokine to maybe treat a certain disease process. For example, interferon alpha used to treat hepatitis B viral infection, but it will also, remember, interferon alpha is also an inflammatory cytokine. So you may just want it to boost the immune response. Remember the type 1 interferons, which are interferon alpha and beta, are very good in your innate immune responses against viral infections. They have an antiviral effect, and we'll cover this more when we're looking at the immune response against viral infections. This we can take advantage of that to try and treat hepatitis B, but the same interferon alpha will produce other effects in the patient, which will make the patient feel sicker, what we call the acute phase response, because they'll produce a sort of inflammatory response. So the same molecule that you're giving for treatment will also make the patient have other symptoms that may make them feel unwell because there's more than one target cell. The other property is antagonism, where you can get two cytokines that inhibit each other, okay? So they both act on the same cell type, but the effects are opposite. So for example, interleukin-4 can cause B cells to class switch from IgM to IgE production, but interferon gamma acting on the same cell, on the B cell, will block this class switching, okay? And then there's cascade induction. This just basically means that they act in sort of a cascade. It's sort of like a cycle, and the best example given here is where you have your activated T helper cells producing, so what happens is first you'll have your macrophage presenting antigen to your T helper cell, and then the T helper cell becomes activated, produces interferon gamma, which comes and acts on the same macrophage to activate it, and then it produces interleukin-12, which will also activate your T helper cell to produce other cytokines, okay? So it's that's sort of act in a sequential fashion so that you get an immune response, okay? Okay, so now we'll move to the nomenclature of cytokines, and we'll just broadly classify them. There are very many cytokines that have been described. It's impossible to know all of them and what they do, but there's some very common ones that we talk about. So probably those are the ones I'd expect you to know. There's no standard naming system that includes all of them, but there are several broad groups. The first one is the interleukins. This is the largest group. And the reason they were called interleukins is because they were thought to act between leukocytes, okay? So white blood cells, but it's been shown that they have effects on many cell types, okay? So the leuc there means basically white blood cells. And just as the name suggests, they're also produced by white blood cells. But remember I mentioned that there are other non-immune cells that can produce cytokines, like your endothelial cells, okay? So, and sometimes even epithelial cells like the ones in the kidneys. So they're usually named with a number. So they're abbreviated as IL. So IL, when you see IL with a dash and then a number, that's basically an interleukin. And the number was basically assigned according to how they were discovered. So the numbers go from 1, 2, and so forth, just in ascending order. The second major group are the interferons. So these ones come from the word, to interfere. So they were named so because they interfere with viral replication, okay? And they're actually very potent activators of the immune system. There are two major groups of interferons, the type 1 and type 2. So the type 1s are the ones that are mainly involved in interfering with viral replication. So that's interferon alpha and interferon beta. Okay, and these are very important. I think there's a group that's actually studying these interferons and people who develop severe SARS-CoV-2 infection. It's been found that there's actually a subset of these patients who have autoantibodies against these interferons. So it means that if they have these antibodies that block the function of these interferons, then they can't mount a good immune response. And these are the patients who end up getting very severe COVID-19 disease. So that just shows the importance of these interferons in the innate immune response against viral infections. The second group is the interferon gamma. Interferon gamma is mainly involved in protection against also intracellular infections. We know viruses cause intracellular infections, but interferon gamma is very important in immune responses against also intracellular bacterial infections, as we'll get to see. And I think you'll even mention this in microbiology when you're talking about the immune response against mycobacterial infections, or basically TB. The third group is basically your tumor necrosis factor. These ones were named so because it was shown that they can induce necrosis. Necrosis is basically cell death in a living host, okay? So it's basically someone is alive, but you get some cells dying, okay? That is necrosis, as you get to define it further in pathology. So tumor necrosis factor was shown to induce necrosis in cancers when injected into animal models at high concentrations, but it doesn't really cause this in vivo. When you get activation of the immune response, you really don't get death of cells. The effects are more subtle. You just get like an acute phase response, for example, induction of fever and other things, okay? And then we have the colony stimulating factors, which are the ones that are mainly involved in production of cells. For example, in hematopoiesis, when you have, for example, your granulocyte macrophage, CSF, or your GCSF, and so forth. So just as the name suggests, these ones are important in production of your immune cells. And then we have the chemokines, which are the ones that stimulate cell migration, okay? So that we call them the chemoattractant cytokines. So when they're produced, they attract different types of cells to the site where they're being produced, okay? So these cells migrate against the gradient of these chemokines towards where the chemokines are. So the chemokines are named based on a number, as you can see here, and spacing of a specific cysteine residue. So you, for example, CCL2, CXCL8, and so forth, okay? And then if the receptor, usually there'd be an R just to indicate that it's a chemokine receptor, okay? And case in point is this other receptor like the CCR5 and CXCR4 that are important in HIV infection. So people who don't have that receptor have been shown to be resistant to HIV infection, or who have a mutation in that receptor. So that was the nomenclature. And then now we'll just talk about the classification. And just as I mentioned, there's no single classification system that would include all the 300 cytokines, but they try to just broadly classify them. So they can be classified based on the structure. So that would be the biochemical structure, or on the function. Okay, I prefer the functional classification. So the structural classification would include the hemopoietin family, and then the interferon family, and then you have the chemokine family and the tumor necrosis factor family, okay? The functional classification, so this is based on the effect of the cytokines. So those that are growth factors. Then you have the pro-inflammatory agents and the anti-inflammatory agents, okay? So I'd actually have to split that into two groups. And then you have the polarizing agents. The polarizing ones are the ones that would basically cause cell differentiation into specialized cells, okay? So this table just shows functional groups of some selected cytokines. And I like this table because it divides them into basically cytokines of the innate immune response and those of the adaptive immune response. And I'll just highlight a few. So for example, interleukin-1 and tumor necrosis factor alpha are produced by innate immune cells, and they act on the cells of the innate immune system. So your monocytes, macrophages, and so forth. So this one's actually pro-inflammatory cytokines. So they're important in inducing inflammation, and inflammation is an innate immune response. And we'll have a whole topic on inflammation. We'll see what it does and how it helps us, and also how it can be harmful. And I think you'll further delve into that in pathology. So they cause what we call the acute phase response, where now patients, they're the ones responsible for causing fever, swelling, tenderness, and all that whenever there's an infection, okay, at a particular site. Interleukin-12 is, as I showed you in that cascade induction picture, it's produced by macrophages, and it influences both adaptive and innate immune responses. Basically activates the natural killer cells to be able to kill, for example, infected cells, but would do the same also to your T helper cells. Interleukin-6 is very important. I just want to highlight interleukin-6, which is produced by macrophages, endothelial cells, and T helper 2 cells. It's also a very potent cytokine of the innate immune response. It causes an acute phase response, also would influence some adaptive immunity. Interleukin-6 is the one that we're seeing. It's the one that is most implicated in the cytokine storm that we see in severe SARS-CoV-2 infection. And you've seen that some patients who have severe infection actually get treated with tocilizumab, which is a monoclonal antibody against the interleukin-6 receptor, okay? Just to try and circumvent this very severe disease caused by SARS-CoV-2. So I just wanted to highlight that because it's quite important. And then I talked about the interferons, which are important in antiviral immunity. So when I talk about cytokines of the innate immunity, it basically means they're produced by the innate immune cells, okay? So your monocytes, macrophages, and so forth. And then you can get even dendritic cells and endothelial cells producing them. The cytokines of the adaptive immune system are mainly the interleukins, like interleukin-2. Interleukin-2 is usually the default cytokine that will be produced after antigen presentation to activate a T cell. Remember, T cells are not able to recognize an antigen directly. It has to be presented to it in the context of MHC. So once you get the peptide of the antigen being presented to the T cell, it becomes activated, produces interleukin-2, which now causes activation of those T cells, proliferation, and so forth, and also can activate B cells. Interleukin-4 and 5 are important in antibody-mediated responses, specifically IgE. So usually they're produced by T helper 2 type of cells. Remember your T helper cells, we have two subsets, T helper 1 and T helper 2. T helper 1 are mainly involved in cellular immunity, and T helper 2 in antibody-mediated immunity. So those ones cause class switching and activation of eosinophils and so forth. Interferon gamma is produced by T helper 1 cells. As I mentioned, it's important in cellular immunity, especially immunity against intracellular bacterial infections, for case in point, tuberculosis and things like that. So what interferon gamma causes, it activates macrophages just to increase their capacity to kill, okay? And then we have transforming growth factor beta. So this one is an anti-inflammatory cytokine, okay? It inhibits proliferation of T cells. So it does the opposite of what interleukin-2 would do, okay? Also inhibits B cell proliferation. This is another table just summarizing the activation and not the activation, the functional classification of these cytokines, and giving some examples of some of the cytokines and what their actual function is. So I've talked about the pro-inflammatory ones. And I'm just highlighting again that it's the cytokines of the innate immune response that are mainly pro-inflammatory. So this group of cytokines are the ones that would cause the acute phase response. So tumor necrosis factor, interleukin-1, interleukin-6, and the chemokines, because remember we talked about chemokines causing cells to move towards where they're being produced. So these are the chemokines are very important in inflammation, okay? And then the anti-inflammatory ones, we've mentioned interleukin, transforming growth factor beta, but there's also interleukin-10. There are those antiviral cytokines which inhibit virus replication. That's interferon alpha and beta. We also call them the type 1 interferons. There are those that activate macrophages, and that's specifically interferon gamma. So interferon gamma is a type 2 interferon. So the differences in this would just, apart from functional, there'd be a slight difference in the structure. And then we have the B cell activating cytokines, so interleukin-4, 5, 6, and 21. And then I think it's even interleukin-13 also activates B cells. Then you have the T cell activating cytokines. Most important is interleukin-2, but interleukin-4 can also do that. So you're seeing pleiotropism of interleukin-4, interleukin-12, and interferon gamma. And then the eosinophil or mast cell activating cytokines, interleukin-3, 4, 13, and 5. These are very important in immunity against helminthic infections and also in allergic immune responses, which we'll come to talk about later. So in terms of secretion, we've said cytokines are secreted by various cell types, mainly cells of the immune system, but you can get some non-immune cells. Most are secreted only when the cells become activated. Remember, we talked about tight regulation because they have very potent effects. So there has to be an inducing stimulus. Most of the time it's an infection, which now causes the cells to secrete the cytokines and so forth. But some are constitutively secreted, so they're always being secreted at, constantly, but at very low levels. But most would only be secreted when there's an inducing stimulus, which usually would be an infection. They're secreted at variable levels, so sometimes it just depends on the strength of the immune response, okay? So that's why you see some patients, for example, with SARS-CoV-2 would get a cytokine storm where they're getting an uninhibited and uncontrolled cytokine secretion, while others are getting just a low-level secretion that doesn't really make them very sick. So for the innate immune system cytokines, usually they're secreted at low levels over a short range. So they tend to basically be secreted to act in either an autocrine or a paracrine fashion. Most of them would just be involved in the inflammatory response. But when there's maybe very a high level of stimulation, you can get these cytokines being secreted at high levels and they act in an endocrine fashion. And when they get secreted in such high levels, you can actually be able to measure them in blood samples, okay? And I've already highlighted which these cytokines are, interleukin-1, 6, and TNF, during the acute phase response. And I talked about interleukin-6 and the fact that we're using a monoclonal antibody against its receptor to block its function in patients with severe SARS-CoV-2 infection. And there are also some centers where they actually, when they're evaluating patients with COVID-19, they actually measure the plasma levels of interleukin-6, okay? For the adaptive immune system cytokines, so they're also secreted at low levels and would have paracrine and autocrine effects. This usually you don't get them, even if you have severe infection, you don't get them being secreted in very large amounts. So it would be very impossible to detect them in fresh samples. You hardly get them acting in an endocrine fashion, okay? So secretion of these cytokines is usually transient. So it would be kind of specific, just in response to infection. And once the infection is resolved, then that stimulus that induced their secretion is no longer there. So secretion usually stops, but we also get, remember, for every system in the body, you have sort of have a balance. You have a way of controlling that system. So we also have cytokines that get produced towards the end of an immune response. Okay? So this just ensure that we stop secretion of this cytokine once the inducing stimulus is no longer there. So the major inhibitory cytokines are interleukin-10 and transforming growth factor beta. And the other important thing with the cytokine secretion is that their receptors are also expressed transiently by activated T cells. This is in the adaptive immune response. So this would prevent inappropriate activation of the immune system. So even if you have, in this case, if you don't get the receptors being expressed, what the receptors of a particular cytokine, even if you have a cytokine being produced erroneously, then it cannot have any effect because it doesn't have receptor. So the production of the cytokine and expression of its receptor goes hand in hand. So you also get, during activation of the genes that code for cytokines, you also get activation of genes that code for their receptors. Remember, the receptors are also cell surface proteins that are actually produced when certain genes are activated. I think this table was just showing cytokines and their general effects. I think you can look at this table later. I just want to highlight, remember I mentioned in the functional classification, we talked about growth factors, okay? One class of cytokines being growth factors. For example, interleukin-7, okay, which is produced by bone marrow stromal cells and acts on lymphoid progenitors to cause them to proliferate. We talked about your colony stimulating factors like GCSF, which is important for neutrophil production, and so forth. Then the other group was the pro-inflammatory cytokines, which I've talked on and on about. And as the name suggests, they're important in an inflammatory response against pathogens. So this would be your TNF, interleukin-1, your chemokines and type 1 interferons and interleukin-6. Then we talked about the anti-inflammatory cytokines, and those ones are interleukin-10 and TGF-beta. Then there was a group I called the polarizing cytokines, okay? So, polarity just means movement towards one direction, okay? So these ones are especially important in the adaptive immune response, where they'll cause differentiation of cells towards a certain phenotype. So for example, for the CD4 positive cells, which are the T helper cells, there are certain cytokines which will cause them to differentiate either into a T helper 1 phenotype, a T helper 2 phenotype, or a T helper 17 phenotype. So the T helper 1 cytokines are interferon gamma and interleukin 12, and these ones are important in an immune response against intracellular pathogens, okay? Usually it would be a cellular immune response. The T helper 2 cytokines polarize your cells towards an antibody secreting phenotype, okay? So these are interleukin 4 and 5. So these are important against, for immunity against helminthic infections. In terms of disease pathology, this T helper 2, if you get uncontrolled T helper 2 cell activation, these are tend to be seen in allergic immune responses, so allergic disease, and also autoimmune disease, where you get a lot of autoantibodies being produced. The T helper 17 cytokines are very important in activating your neutrophils, especially, and causing an immune response against basically inflammatory responses where you get your neutrophils being involved. So extracellular bacterial infections and also fungal infections. I talked about the fact that cytokines act by binding to their cognate receptors. There are different types of receptors that are classified based on their structure. So there are 6 major cytokine receptor families, and this is just a diagram to highlight how they look. So you have your immunoglobulin family receptor, your hemopoietin receptors, or cytokine receptor superfamily, where you have an alpha, beta, and a gamma chain. You have the interferon type of receptors, which usually would have your JAK signaling molecules there. You have the tumor necrosis factor receptors, interleukin 17 receptors, and chemokine receptors. And we said that once the cytokine binds to its cognate receptor, then it induces the signaling, intracellular signal transduction molecules. And remember, I mentioned that most of the cytokine receptors are not expressed on high numbers on resting cells, but once the cell is activated, then you get, not only do you get production of the cytokine, but you also get upregulation of the cytokine receptors. So you get more of the cytokine receptors being expressed. And then once you get the cytokine binding to its receptor, you get them aggregating at the surface of the cell. Basically, they colocalize, you get them moving towards each other just to enhance the intracellular signal transduction molecules as shown in this picture. So for example, you get these 2 chains of this receptor colocalizing or moving towards each other, and more of other cell surface receptors. And I think you've talked about intracellular signal transduction molecules in physiology, which basically is what aids in moving this signal to the nucleus so that you can get specific gene transcription, okay? So the net effect of cytokine binding on its receptor is to cause or change gene expression of the target cell, okay? So what are the roles of the cytokines in the immune response? So one is initiation of inflammation. There's also communication, and so usually it's the innate immune system cytokines that would cause that. communication between innate and adaptive immune systems, like we've seen some of the cytokines, for example, produced by macrophages, which act on T helper cells during antigen presentation, okay? Then you have T cell priming, okay? So activation of your T cell or differentiation of your T cell. So that will be development of T cell specialization. So those would be the polarizing cytokines. And then, of course, winding down of the immune response. So this is where you have your inhibitory or anti-inflammatory cytokines playing a role. This is just reiterating. I've talked about the cytokines of the innate immune response. So these are usually secreted by macrophages and monocytes. You could also have your dendritic cells and endothelial cells. And they are pro-inflammatory cytokines, specifically interleukin-1, tumor necrosis factor, and interleukin-6, and then chemokines, which now recruit neutrophils to extravasate from the blood into the site of an infection. And so these are what would cause acute inflammation. The other innate immune response cytokines are the type 1 interferons, the interferon alpha and interferon beta. So these ones are produced by virally infected cells. Once you have a virus gaining entry into a cell, it starts producing the type 1 interferons, which now act in a juxtacrine or a paracrine fashion on neighboring cells. And basically what they try to do, not only do they try and inhibit viral replication, but they make these neighboring cells more resistant to viral infection. And they also cause these cells to be susceptible to killing, just to try and control infection or the spread of the viral infection, okay? So in T cell priming, this is where you're getting now activation of your T cells. So in antigen presentation, you know that the first signal to activate your T cell would be your antigen presenting cell expressing MHC, and onto that MHC you have your peptide antigen being carried. So that is what is being presented to your T cell through the T cell receptor. That is the first signal. That is not enough to activate your T cell. You need a second signal. So that second signal is what we call the co-stimulatory signal, okay? So for example, CD40 would be a cytokine that is expressed, not CD40 ligand would be a cytokine that is expressed on a surface of a cell to bind to CD40 on these cells so that they're activated. The other would be the B7 family of molecules like CD80, CD86, okay? So the other co-stimulatory cytokines that can be expressed that are soluble, like interleukin-1 and type 1 interferons, they're also important in initiating T cell responses. We've talked about upregulation of the receptors. So for example, when T cell is activated and produces interleukin-2 to act either in an autocrine or a paracrine effect, you also need upregulation of the interleukin-2 receptor, okay? I've talked about the polarizing cytokines. So this would be the ones that are involved in specialized T cell responses, the T helper 1, T helper 2, and T helper 17. And I mentioned that the T helper 1 responses are important in an adaptive immune response against intracellular pathogens. So the cytokines involved here are interleukin-12 and the type 1 interferon. So the macrophages or the antigen presenting cells, as they're presenting the antigen to the T cells, they secrete interleukin-12. Once a T cell is activated, it produces interferon gamma. And so this now T cell that produces interferon gamma, we call it a T helper 1 cell. And the interferon gamma now comes and acts on these macrophages to activate them, okay? Just to enhance the killing of these intracellular pathogens, and they also favor production of IgG by B cells, okay? But the main thing is that you have a cellular immune response where you have activation of your T helper 1 cells. For the T helper 2 responses, I talked about these ones being important in the immune response against helminths. Okay, so they change basically what leads to development or change in this phenotype is the gene transcription. So you get a different transcription factor being activated that now switches these T helper 2 cells to favor IgE production, okay? So usually they'll produce interleukin-4. These T helper 2 cells produce interleukin-4 that activate B cells towards production of IgE. And they also produce interleukin-5, which activates mast cells and eosinophils, which are also important in anti-helminthic responses. Remember, the granules that the eosinophils carry contain enzymes that are very toxic to helminths. They also produce a chemokine called eotaxin that would be important for attraction of eosinophils to the site where the helminthic infection is. And then there's the T helper 17 responses, which are important in immune response against extracellular pathogens, so mainly bacteria and fungi. So these pathogens, usually when you get your innate immune cells being activated, they produce interleukin-23, which is the one that will act on your T helper cells to cause them to differentiate into T helper 17 cells. And T helper 17 cells secrete interleukin-17, and that's why they were named T helper 17, which is the one that attracts neutrophils to the site of infection, where you have your bacteria or your fungi. And what do the neutrophils do? You know, they're phagocytes, okay? We also have this polarizing cytokines, TGF-beta. I know we said it's an anti-inflammatory cytokine, but it's been shown to be very important in gut immunity, so mucosal immunity within the gastrointestinal tract. It causes the lymphoid cells within the mucosa-associated lymphoid tissue to class switch from IgM to IgA, and then you get that IgA being secreted into the mucosa, so of the GIT. I've already mentioned the anti-inflammatory effect, but I just wanted to highlight that TGF-beta is important in causing class switching within the mouth, so that you get IgA being produced and secreted within the gut mucosa. The other type of T cell is a T regulatory cell, so also called TREGs, okay? These ones are, as the name suggests, they are important in regulation of the immune response, okay? Sort of suppressing excessive immune responses. So they're important in peripheral tolerance and preventing responses against self-antigen. So these TREGs are the ones that produce the anti-inflammatory cytokines, interleukin-10 and transforming growth factor beta. And then the role of cytokines in ending the immune response. Of course, this will be mainly now your TREGs producing the anti-inflammatory cytokines. And there are certain things that favor ending of the immune response. Of course, we said that for cytokines to be produced, usually there has to be a stimulating antigen or an inducer, and that would be the infection. So as the immune response continues, you get the antigen being cleared. So as you have less and less antigen, you get stimulation of your T cells declining. So you get lower levels of cytokine production and less or downregulation of receptor expression. So, and then you get now your anti-inflammatory cytokines being produced, which inhibit T cell proliferation, okay? Another important thing you'll see when we talk about tumor immunology in year 4 is that sometimes you get tumors producing anti-inflammatory cytokines, which favors growth of the tumors, very important in cancer, so that now your immune system is not able to get rid of the cancer. And then on cytokine-related diseases, so because cytokines are very potent, sometimes they may cause injury. For example, when you have so sometimes when you get excessive cytokine production, you end up getting excessive inflammation that injures host tissues. For example, with certain bacterial toxins, and I'm sure you've talked about this in microbiology. For example, the toxic shock syndrome toxin seen with staphylococcal infections, or even staphylococcal enterotoxin, where you get cytokines activating a large number of T cells, which ends up causing excessive inflammation. This is another scenario where you can get a sort of cytokine storm when you have two. So this interferon gamma activates too many macrophages, and the macrophages now keep producing pro-inflammatory cytokines. Okay, so same thing that we see in severe SARS-CoV-2 infection. So what the cytokine storm does, remember we said this interleukin-1, interleukin-6, and TNF cause an acute phase response, okay? And when you have too much of this being produced, then you cause a picture that looks like sepsis, okay? With shock and multi-organ failure and disseminated intravascular coagulation. So this is what leads to severe disease, this septic symptoms, okay? So seen in bacterial infections, but can also be seen in viral infections, for example, with SARS-CoV-2, and even in influenza infections in some cases, okay? There are situations where there can be disease caused because there's a mutation in the gene that codes for the cytokine or its receptor. Okay, so for example, the interleukin-2 gamma chain receptor, if there's a mutation in that, then you get a receptor that's not functional, okay? So it causes an immune deficiency because even if interleukin-2 is being produced, the receptor is not functional, okay? And because it's a common gamma chain, this same gamma chain is shared by the interleukin-4 receptor, 7, 9, 15. They all share that gamma chain. So the function of all these cytokines will be affected, okay? So usually what happens, remember we said interleukin-7 is important in activating your lymphoid progenitor cells to develop, okay? So you don't get T and B cell development, or even if you get some, they're not going to be functional. So these patients who have this mutation tend to develop what we call severe combined immunodeficiency. We'll talk about this later when we're talking about primary immunodeficiencies. The other cytokine where you can have loss of function is when there's a deficiency in CD40 ligand, okay? Remember we said this can be it's a cell-expressed cytokine as opposed to the soluble ones, and it acts as a costimulatory signal with CD40 on your B cell. So this is usually expressed on T cells. So if you have B cell presenting antigen to T cell, the second molecule, so the first molecule will be the MHC on the B cell interacting with the T cell receptor. The costimulatory molecule will be the CD40 ligand interacting with CD40. And this second molecule is what is important for class switching of your B cells from IgM production to either IgA or IgG or IgE. So if there's a mutation in this CD40 ligand or it's missing, then you cannot get class switching of your B cells. So these patients only produce IgM. So they tend to have very high concentrations of IgM and very low concentrations, or actually absent IgG, E, or A. So they basically don't have memory B cells being formed, okay? The other one is the Fas ligand. Remember we said Fas ligand is also a cell surface expressed cytokine. And when it interacts with Fas, it's important in basically causing apoptosis of cells, for example, removing excessive lymphocytes after immune response. So patients who do not have the Fas ligand tend to have too much accumulation of the lymphoid cells, and especially the autoreactive lymphocytes during T cell development. So they tend to develop a disease called autoimmune lymphoproliferative syndrome. So it's a lymphoma-like syndrome, but they also have autoimmune effects. I talked about chemokine receptors being important in HIV infection, so cytokines and infectious disease. So remember, I would say the chemokines are named either CXC or CCR, depending on where a certain cysteine residue is, and then R would mean a receptor. So there are these two receptors, CXCR4 and CCR5, that serve as co-receptors for HIV infection. So the main receptor is CD4, and then you have this co-receptor. So these tend to colocalize with the CD4 to allow entry of HIV. So patients who have a mutation in the CCR5 have been shown to be resistant to HIV infection, okay? mainly seen in Caucasians, I think. In terms of assays, I think I'm getting towards the end. I know time is running out. We can, in some scenarios, measure or quantify the amount of cytokines in biological specimens, for example, in biopsies or cells or body fluids, but this is not done routinely. And as I mentioned, for the cytokines or the adaptive immune response, they're produced in very low quantities, so it will be difficult to measure that. But this can be applied in cases where you have a lot of cytokines being produced in the innate immune response, like interleukin 6 or tumor necrosis factor. So where you have like toxic kind of presentation because of excessive cytokines. And there are several platforms that can be used to measure them. We'll talk more about these platforms in year 4, so I wouldn't even go into details of that. In terms of therapy, I'll just mention that you can either use like recombinant cytokines, either in situations where you want to stimulate or enhance an immune response. So for example, that would be called immunotherapy. So for example, in cancer patients, you can give interleukin 2 to increase proliferation of your T cells, interleukin 4 to increase B cell activation, interferon gamma to increase antigen presentation, interleukin 12 to activate natural killer cells. We don't have these agents for this kind of immunotherapy in the country, but I guess the best example I can give is use of Neupogen. Neupogen is granulocyte colony stimulating factor, which we give in neutropenic patients. So patients who have neutropenia due to chemotherapy, as long as it's not an acute myeloid leukemia. So just to increase their neutrophil counts, because also you don't want them to be neutropenic because now they become susceptible to infection. So here you're enhancing or stimulating an immune response. You can use cytokines to block or suppress immune responses. So that would be cytokine blockade. For example, we give transplant patients an antibody against CD25, which is the interleukin 2 receptor, so that even if they're getting like rejection responses and their T cells are being activated to produce interleukin 2, you block the receptor so that they don't have any effect of these activated cytokines. And then I talked about anti-TNF used in rheumatoid arthritis patients. So these are just some examples of cytokine-based therapies. I've also talked about Rituximab, which is an antibody against interleukin 6 that we're using for patients with severe SARS-CoV-2 infection. And that is the end. Do you have any questions? Any questions?